Good afternoon. I would like to thank the organizers for their kind invitation. There is a clear link between small fiber neuropathy and fibromyalgia. On the other hand, most people will agree that fibromyalgia is a stress-related disorder. So I do believe that the key issue in fibromyalgia research is to find out how different stressors become neuropathic pain and where different stressors become neuropathic pain. So what I plan to do is to talk about fibromyalgia as a stress-related disorder, to discuss stress response system, particularly sympathetic dysfunction in fibromyalgia, and to talk about our long-standing proposal of fibromyalgia as a sympathetically maintained neuropathic pain syndrome. To talk about the clear relationship between small fiber neuropathy and fibromyalgia, and finally to discuss the key question, how and where stress becomes neuropathic pain in fibromyalgia. Most people will agree that fibromyalgia is a stress-related disorder. Control studies have shown that different stressors, such as childhood maltreatment, violence, physical injuries, infective agents, and autoimmune illnesses are clearly linked to fibromyalgia. We have studied in different ways sympathetic malfunction in fibromyalgia using diverse heart rate variability analysis, and we found changes consistent with ongoing sympathetic hyperactivity associated to sympathetic hyporeactivity to stress. Our genetic studies of key autonomic components have also shown a difference between fibromyalgia patients and controls. Validated questionnaires show that fibromyalgia patients have many dysautonomia symptoms, contrary to what happens normally. Fibromyalgia patients have norepinephrine-evoked pain, as shown in this slide, where we compare fibromyalgia with rheumatoid arthritis and healthy controls. In a double-blind fashion, we injected minute amounts of norepinephrine or placebo in the 4 arms, and fibromyalgia patients have norepinephrine-evoked pain. Dysautonomia explains the multiplicity of fibromyalgia symptoms as no other model. We have also long proposed that the pain in fibromyalgia is of neuropathic origin based on these issues. It is a stimulus-independent chronic pain state accompanied by allodynia and paresthesias. This proposal has been backed in the last decade by a description of small fiber neuropathy in fibromyalgia. As you know, small fiber neuropathy is a disease of the thin nociceptive fibers innervating skin and internal organs. It is associated to diverse metabolic and immunological illnesses. And very important, the idiopathic form is linked to NAV17 sodium channel gene mutation. Clinical features are similar to fibromyalgia, pain, paresthesias, and autonomic dysfunction. The gold standard for the diagnosis is skin biopsy showing skin denervation. And here, this list is by no means complete and shows how different groups of investigators found small fiber neuropathy in fibromyalgia. We use corneal confocal microscopy in view of the fact that the cornea receives the most dense small fiber innervation of the body. So this confocal microscopy is a virtual small nerve fiber biopsy. In this study, published in 2015, we compared 17 fibromyalgia patients with 17 healthy controls. And what we found is thinner and fewer small nerve fibers in the cornea of fibromyalgia patients. These results have been corroborated by other investigators. And these slides show how corneal confocal microscopy looks, and you can see the difference between a patient and a healthy control. In this recently published study, we attempt a clinical pathological correlation. And what we found is that those fibromyalgia patients without severe anxiety or depression, and this anxiety or depression was rated according with validated questionnaires. And what we found was a very strong correlation between corneal denervation and small fiber neuropathy symptoms. So this suggests that corneal confocal microscopy can become a very useful test in the diagnosis and follow-up of fibromyalgia patients. Our main proposal is that this marvelous structure, dorsal root ganglia, are the main fibromyalgia pain source. Humans have 30 pairs of dorsal root ganglia lying along the spine, as shown here. Each ganglion houses approximately 100,000 small nerve fiber nuclei that are enveloped by immune competent glial cells. And dorsal root ganglia are in close contact, direct contact with the paravertebral sympathetic chain. Dorsal root ganglia have a unique anatomy and physiology. Embryologically, they belong to the central nervous system. During development, they are extruded to the peripheral, but remain covered by meningeal layers and by cerebrospinal fluid. Nevertheless, they lie outside the blood-brain barrier. So different molecules, pro-nociceptive molecules, infective agents or immune complexes can gain direct access to the dorsal root ganglia. Furthermore, dorsal root ganglia have a unique pseudo-unipolar configuration. From the soma, axon exits and divides in a T-shaped junction. So, incoming painful stimuli from the peripheral can be filtered at the dorsal root ganglia, and more important, dorsal root ganglia are able to generate painful stimuli. Dorsal root ganglia have a rich nociceptive immune arsenal with different type of ion channels, calcitonin gene-related peptides, substance P, glutamate, and nerve growth factor. All of these very potent pain-inducing substances, as well as immune-competent perinuclear glial cells, and also macrophages, lymphocytes populate dorsal root ganglia. And a recent discovery shows that dorsal root ganglia are able to sequester antigen-specific antibodies. So in this ganglia, an immune pain-inducing short circuit can be established. And here, ladies and gentlemen, let me introduce to you what I consider the anatomical structure of this decade, the dorsal root ganglia. In the left side, we have the small nerve fiber nuclei, you can see here, tightly enveloped by immune-competent glial cells. There is rich blood supply to the dorsal root ganglia. Macrophages, lymphocytes populate this ganglia, and this is a peculiar pseudo-unipolar configuration. The animal model is very clear. There is a stress-evoked dorsal root ganglia inflammation that leads to hyperalgesia. And this stress could be environmental or psychological distress, such as unpredictable noise or water caging, induces dorsal root ganglia inflammation and also hyperalgesia. Typically, physical stress provokes dorsal root ganglia sympathetic sprouting, establishing abnormal connections between the sympathetic nervous system and the pain pathways. And this is the basis of the sympathetically maintained pain, as you can see here. Normally, dorsal root ganglia don't have sympathetic innervation, but after trauma, things change completely. And norepinephrine induces pain. As already told, they can sequester antigen-specific antibodies. In these days, it's impossible not to talk about SARS-CoV-2. SARS-CoV-2 receptors are broadly expressed in human dorsal root ganglia neurons, and this could be the basis of the long COVID syndrome. And there is unfolding clinical evidence linking dorsal root ganglia to fibromyalgia. A strong argument is the clear link between small fiber neuropathy and fibromyalgia. The most important part of the neuron, the nuclear or the soma of the small nerve, is within the dorsal root ganglia. We describe severe fibromyalgia associated to particular dorsal root ganglia NaV17 sodium channel genotype. In fibromyalgia patients, exercise increase dorsal root ganglia pro-nociceptive receptors, hypothetically explaining the exercise intolerance that these patients may have. IgG from fibromyalgia patients induces mice hyperalgesia, and it labels precisely and specifically dorsal root ganglia, not the brain, not the spinal column. And the few post-mortem inquests have found dorsal root ganglia inflammation. So in this scheme, we summarize our proposal. We have a stress-related illnesses, and we propose that due to this stressor, there is dorsal root ganglia phenotypic changes with nociceptive nuclear degeneration. This alteration will produce the peripheral denervation, increase ion channels, sympathetic sprouting, increase glial cell activation, neuropeptides, and neurotrophins. The pathogenesis, the proposed pathogenesis is small fiber neuropathy and dysautonomia, with the resulting clinical features of widespread pain, paresthesias, allodynia, and other multi-system features. In conclusion, fibromyalgia is a stress-related disorder. There is a stress response system malfunction, and this explains fibromyalgia multiple symptoms. There is a clear link between small fiber neuropathy and fibromyalgia. Dorsal root ganglia house small nerve fiber nuclei surrounded by immune competent cells and by pain transmitting mediators. Psychological, physical, infectious, and/or autoimmune stressors may induce dorsal root ganglia inflammation with the resulting hyperalgesia. Unfolding clinical evidence links fibromyalgia to dorsal root ganglia inflammation. And this is our main proposal. Dorsal root ganglia are the key sites where diverse fibromyalgia driving stressors, including psychological distress, become neuropathic pain. What are the future directions? To search for dorsal root ganglia inflammation in fibromyalgia using advanced neuroimaging or dorsal root ganglia culture. And we are working on that. To continue with postmortem inquest, and there is this open different therapeutic possibility. Dorsal root ganglia ion channels and neuropeptides are attractive targets for analgesic development. I thank you for your attention.